Welcome to Brain Ponderings. I'm the host, Mark Matson. This will be the first in a short series of presentations on brain bioenergetics. And that has to do with, as its name implies, how the brain cells acquire, utilize energy. It's actually very complicated in the brain, in part because the cells are very complicated in the brain in terms of their structure and functionality. And as a part of this, I will talk about this from an evolutionary perspective, as well as getting into some details on how nutrients in food end up generating ATP in brain cells. And then I will talk about how mitochondria uh, are moved around within neurons, how they divide, how they can fuse with each other, and how when they become dysfunctional, they're removed. Then I'm going to talk about something we've worked on a lot in my lab over the years, how fasting and physical exercise affect the brain. So how bioenergetic challenges, the challenge of not having food intake, or the challenge of vigorous expenditure of energy affects the brain. It turns out it's very interesting. There's what are called fastokines and exerkines that are released from various organs into the blood, and they act on the brain in interesting ways. And then in the last presentation, I'll talk about brain bioenergetics from the perspective of aging and neurodegenerative disorders. So this first presentation is going to be an overview uh, from an evolutionary perspective and from a, I guess, kind of a biochemistry practical viewpoint. Okay, the first question uh, that comes is, where does the energy come from that fuels the nerve cells and other cells in our brain? Well, ultimately it comes from sunlight, from the ultraviolet light, uh, the energy in that ultraviolet light being used in photosynthesis in the chloroplasts of plant cells, and that in that process of photosynthesis, carbon dioxide, carbon is taken from the air and used to synthesize glucose, as well as the fats and proteins in plants. And then we consume the plants, or other animals consume the plants and we consume them. And so this carbon that is the backbone of all molecules in us and other living organisms uh, comes from the carbon dioxide in the air. So this slide shows this cycle. And by the way, if you're listening on Spotify or other audio outlets, I am showing slides on my YouTube channel, Brain Pondering's YouTube channel. So you might want to, if you have time and are in a situation to do so, look at these or come back later and look at them again. Right, so this diagram shows this carbon cycle. Okay, our brains, human brains, have billions of neurons and trillions of synaptic connections between the neurons. Moreover, there are as many or maybe a little more non-neuronal cells in the brain, that is glial cells, the astrocytes, the myelinating oligodendrocytes, and the microglia, the immune cells in the brain. And over the last couple of years in various podcasts, we've talked about these glial cells. Now, nerve cells are excitable and similar to our muscle cells, skeletal muscle cells, our cardiac cells. They require a lot of energy to maintain and restore the charge potential across their membranes. and that comes in the form of ATP, adenosine triphosphate, and there's energy in the bonds of ATP that can be utilized to drive various reactions and the function of various proteins. And a little later, I'll talk about the importance of ion gradients across neurons in their function and the role of ATP in that. Now, neurons also have elaborate morphologies. That is, they are often very long. They have a cell body. They have often a long axon that can extend distances in humans up to a meter, a yard. For example, in the motor neurons go from our spinal cord down to the muscles in our that control our foot movements, or even in the upper motor neurons in the brain that go from your brain to your spinal cord. Okay, so these cells are large with elaborate morphologies, and they have to be able to have energy throughout their axon, their dendrites, which are often branching. And as most of you know, the mitochondria in the cells are the source of ATP. So they're the main energy factory in the cells. And these mitochondria have to be distributed throughout the length of the axon and its branches and dendrites, because energy is needed throughout the cell, essentially in real time. And then I'll talk later about how these mitochondria, they can divide, so they can increase in number in cells. They can move. They are moved in a controlled manner within the arbors, the dendritic arbors and axons of the cells. Okay, so where do mitochondria come from? There's pretty compelling evidence that during evolution, they initially rose in a symbiotic relationship between bacteria or bacteria-like organisms, which are actually thought to be the first forms of life on Earth. So a symbiotic relationship between those and another cell that didn't take up these bacteria. So bacteria, of course, they have to have energy. They take glucose and they convert it to ATP. And it was apparently to the advantage of some primordial, what we call eukaryotic cell, to, I guess maybe even they were infected or endocytosed, these bacteria-like cells, and at some point, the cells were selected for that retain these bacteria-like cells within them. And then over millions and millions, billions actually of years of evolution, actually the functionality of what we call mitochondria changed, became improved in terms of efficiently generating ATP, and I'll talk about that. And also, interestingly, they have other functions in cells, including neurons, which I'll talk about. Okay, so evolution of the brain. What was the driving force for brain evolution? Well, there's probably multiple ones. Survival and reproduction are kind of the main things that organisms need to do. They need to survive to reproduce. They need to reproduce to pass their genes on to the next generation. And there's pretty strong evidence that one, or perhaps the most important driving force for brain evolution, was food scarcity. Competition for limited availability of food, which was often sporadically available, not continuously available. So we and a lot of organisms are adapted to going extended time periods with no or very little energy intake food intake. There's some very nice work from Richard Passingham and Steve Wise, where they're studying foraging in non-human primates. So our boreal ancestors, that is, non-human primates who lived mostly in the canopies of trees. And of course, they were adapted to that, moving around efficiently. And they have very good visual systems. They have pretty good sense of smell. And Passingham and Wise did some really neat experiments where they essentially showed or provided strong evidence that the prefrontal cortex, the region of our brain and non-human primate ancestors, our cousins, that plays important roles in decision making, in suppression of impulses, and in complex cognitive decisions. So thinking, making a decision, then acting. And this brain region, prefrontal cortex, has expanded dramatically during primate evolution, and it's the biggest in humans of any mammalian species. Okay, so they provided evidence that neural circuits in the prefrontal cortex are very critical for decision making and in terms of foraging. So deciding when to leave one area where food resources are becoming scarce because you're consuming them, and then going to another. So that's pretty interesting. Seems to be an important role for food scarcity and even most recent evolution of non-human primates and humans. In fact, the increase in size of the brain during hominid evolution corresponds with things like abilities like being able to make tools, initially like flake stones, spears, etc., for hunting, for processing food, cracking nuts, and so on. The harnessing of fire, so we could cook meat and so on. So this shows the cranial volumes, the skull volume increased a lot in the last few 100,000 years of human evolution. Or sorry, million years. Okay. And we know from archaeology and anthropological information that all the early tools invented by humans had to do, as I said, with processing or acquiring food. This continued even with the agricultural revolution, the domestication of plants and animals. So that was a major advance. We could use these animals as shown here to enhance our ability to cultivate the domesticated grain crops like wheat, right, soy, rice, and so on, corn. And even you can make the argument that, even inventing things like internal combustion engine and complex machinery, a lot of that early machinery invented by humans had to do with acquiring, processing, or transporting food. And of course, nowadays, we don't spend our time thinking about how to get food, and we don't spend a lot of time moving through our environment, expending physical exertion to acquire food. My own view is that may not be so good and may be contributing to some adverse effects on the brain, and we'll talk about that later. Okay, and one can even make the case that artistic expression was initially motivated by food scarcity. At least the earliest ones that have been found, cave drawings, were drawings of animals that we consume or otherwise use in relation to food. So that's kind of interesting. And not shown here is some of these have like even scenes of these hunter-gatherers hunting or harvesting food. Okay, interestingly, there's evidence, and here's an example from Ian Tattersall's a nice paper he had in 2023, where he essentially made the case that the size of the human brain has decreased since prior to the agricultural revolution anyway. That's based on skull volume. So that's shown here where the contemporary human brain is a couple hundred milliliters smaller than our ancestors that had to spend a lot of time thinking and moving. So maybe we're not using some of the brain regions. Clearly, we can function very well in doing specialized tasks. So we're becoming more specialized, and maybe our brains are being evolving accordingly. Okay, related to this, and I'm almost done with the evolutionary perspective, is that there's pretty good evidence that individuals with obesity, be they humans, even human children or adolescents, or some pets, these dogs actually look, maybe they're not overfed too much, but a lot of people overfeed their pets. That's kind of understandable because they keep bugging you if you don't feed them, or some of them do. But anyway, for example, in the case of dogs, so the domestication of dogs has led to a decrease in the overall brain size. And that's based on comparing the brain size of wolves in the wild, which all the domesticated dogs descended from through intelligent design by humans. So that's like one example of intelligent design by humans. Humans do the intelligent design. Right, so domesticated species with the same body size as a wild wolf have smaller brains than the wolf. And maybe it's because they don't have to use their brain a lot and haven't, as they've been selected for over generations and generations of selection by humans. Also, measurements of brain size and particularly certain brain regions by MRI imaging of the brain indicate, and there's many studies on this now, that people with obesity, including children with obesity, have smaller size of hippocampus and a couple other brain regions that are critical for learning and memory and cognitive function compared to age-matched, healthy weight individuals. And when I talk about the effects of exercise and fasting on the brain, that will make sense. Also, interesting from an evolutionary perspective is that animal studies have shown that when you essentially maintain rats or mice on extremely low calorie intake, so they're like right on the point of starvation, the size of all their organs, the liver, even the heart, muscle, and these are animals that aren't exercising, and so on, decrease except for the brain. So the brain is preserved under conditions of extreme food scarcity. That kind of makes sense because it's like so critical. You're never going to be able to get food if your brain isn't working. Your liver can decrease in size and still carry out its function as well as other organ systems. Okay, so now I'm done with the evolutionary perspective. And now I want to talk a little bit about uh, cellular metabolism in the brain. Okay, so here's some pretty images of neurons in the human brain, of which, since I said, there's billions, and this is showing these long axons extending throughout from one region of the brain to the other. All right, so turns out there's pretty much a direct correspondence between the amount of activity in a particular brain region, how fast the neurons are firing, how many are firing, and the blood flow to that brain region. And that's essentially similar to what happens in skeletal muscle cells during physical exercise. Blood flow to the muscle increase because the cells need energy from the glucose or fats or ketones in the blood to keep them running. Same is true for the heart. When you're running real hard, there's increased blood flow to the heart. Same happens in the brain. And in fact, the way that functional magnetic resonance imaging works, that is a method whereby you can, using MRI, uh, uh, get an indirect indicator of neural network activity throughout the brain. The functional MRI images are actually what you're getting as a reflection of blood flow. And since there's a tight coupling of neuronal activity to blood flow, there's essentially, you get a good indication of how much activity is going on in a certain brain region at a certain time, and you can have the subject in the MRI scanner do a cognitive task, or you can show them whatever horror film. It's almost Halloween now, and blood flow will change, whether they're thinking about something or afraid of something, and the brain regions that seem to be important in responding to those factors. Okay, so anyway, my point here is when you're measuring blood flow, you're looking at, when you're doing MRI functional imaging, which measures blood flow, you're getting indirect measure of neural network activity and therefore energy production and utilization in neurons, because the more active they are, the more ATP they produce. Okay, so throughout the brain, most of the neurons use the excitatory neurotransmitter glutamate. And so 90% of the neurons in the brain use glutamate. I talked about this when I was kind of give a preview of my book, which is out now, Sculptor and Destroyer, about glutamate. And then about maybe 15% of the neurons, maybe about 8% of the neurons use the inhibitory neurotransmitter GABA. So throughout the brain, the neural circuitry is excitatory glutamatergic neurons, inhibitory GABAergic neurons. The other neurotransmitters, serotonin, dopamine, norepinephrine, acetylcholine, they only comprise about 2% of the neurons. And the only way they modify brain function, behavior, or thinking, anything we do or sense, the only way these other neurotransmitters affect those is by modifying the activity of the glutamatergic neurons. The GABAergic neurons, one of their main function is to prevent excessive activation of glutamatergic neurons as occurs in epilepsy. So some of the main treatments for epilepsy reduce excitability of the glutamatergic neurons, either by directly affecting them or by enhancing the GABA neuron inhibitory activity. In fact, this is how fasting and ketones suppress seizures, is by increasing the GABA inhibitory activity and making sure these excitatory neurons only fire when they need to mediate their behaviors. Okay, so I mentioned this coupling of neural activity to blood flow. So So this is a neuron here. This is a glial cell and astrocyte. This is a blood vessel here with a vascular endothelial cells, and these are what are called pericytes. And then this is a vascular smooth muscle cell, which can contract and constrict blood flow or relax to increase blood flow. And one way in which neural activity increases blood flow is through nitric oxide. There was a Nobel Prize actually given for the discovery of nitric oxide in the vascular system, where it was shown to be released from these vascular endothelial cells and acts on the muscle cells in the blood vessels and relaxes them. So you get increased blood flow. And then there's a role for astrocytes too. I'm not going to go into detail on this in this what's called neurovascular coupling. And you know, in most of my slides I give a reference, so you can on PubMed find these articles, and they're mostly review articles. So if you want to know more about that, you can go to that reference. This is from an article Simo Camandola and I wrote a review article on brain energy metabolism a while ago. And this just kind of expands this. And kind of the key thing I want to talk about here is what are the nutrients, the molecules in the blood that our brain cells use for energy. Most of you know this already. Glucose from carbohydrates. So when we eat carbohydrates, they're broken down to glucose. The glucose can be stored as glycogen in the liver, but it circulates in the blood. It can be actively moved by glucose transporters out of the blood into cells outside the blood in the tissues, be they in the brain neurons or glial cells. Okay, so that's one energy source for cells is glucose. Another energy source is fats or lipids, fatty acids. We generally, in our fat cells, we store triglycerides. Many of you have heard about them, diglycerides. And those are stored in your fat cells. And then when you fast for an extended time period, like 12 to 14 hours, then those die and triglycerides are broken down to what are called short carbon chain fatty acids, free fatty acids, and they're actively moved into cells. But they're also the precursor for the ketones that are produced in liver cells. So, and I'll talk about that in a minute. So these are the main sources: that glucose, fats, and ketones derived from fats, which are a very efficient energy source. Okay, and one point of the slide is that in the membranes of cells, there are proteins that recognize and move glucose, fatty acids, and ketones into cells. So this is an active process, and these proteins have evolved over long periods of evolution. In fact, there's different but functionally very similar glucose transporters in neurons compared to astrocytes, compared to vascular endothelial cells, for example, and different transporters called MCTs, monocarboxylic acid transporters for the ketones. Interestingly, those MCTs, not only they transport ketones, but they transport lactic acid, lactate, which is something you may have heard about if you're an athlete, a lactic acid threshold where you get to a certain point and you start to go anaerobic. All right, so how is glucose used to produce ATP? There's a process called glycolysis, okay, which doesn't involve the mitochondria. And this occurs in the bacteria I mentioned a long time ago. And so this is the pathway. I'm not going to go into this. There's enzymes that act on glucose to break it down into smaller molecules, and during this process, ATP is produced. But actually, it's not very efficient. So it actually takes 2 molecules of ATP to actually break down the glucose to generate ATP, and then 4 molecules of ATP are generated here. So we only get, for each molecule of glucose, only get net 2 ATPs generated in cells. Okay, so during evolution, a more efficient system for generation of ATP from glucose was established, and that involves the so-called TCA cycle and the electron transport chain in the mitochondria. So the TCA, or Krebs cycle, Hans Krebs discovered this. What happens is that, oh, sorry. Right. So the bottom line is that the glucose is broken down. I'm sorry. Right. The glucose is broken down into pyruvate, which then broken down to acetyl-CoA. This enters the TCA cycle. Okay, so you get pyruvate from glycolysis, for example. And it's broken down to acetyl-CoA. I should also say that fatty acids and ketones through a different pathway end up going to acetyl coenzyme A, which enters the Krebs cycle. And during this process, there's a reduction of NAD plus nicotinamide adenine dinucleotide to NADH. And this NADH is used in the electron transport chain. So the TCA cycle is in the mitochondria. It uses pyruvate to generate acetyl-CoA, and that's used to reduce NAD. Okay, and then this NAD, reduced NAD, and also FAD, flavin adenine dinucleotide, are used in 2 protein complexes in the electron transport chain. So the electron transport chain is several groups of proteins that function to move hydrogen across the inner membrane of the mitochondria from the inside the mitochondrial matrix, the inside of the mitochondria, to the outside of the inner membrane, the intermembrane space. Okay, so you get a hydrogen ion gradient, and then that hydrogen ion gradient is used to generate ATP in complex 5 of the electron transport chain. So there's complexes 1, 2, 3, 4, and then 5. So these first 4 ones are involved in moving the hydrogen ions out, generating a gradient, and then the energy generated by that electrical gradient is used to convert ADP to ATP. So, and this phosphate, then you get this bond, and that bond can be used by various proteins to promote their function. Okay, also shown here is, and this is very efficient, this oxidative phosphorylation. So 1 glucose molecule is used to generate 32 ATP if it goes through the TCA cycle and the electron transport chain. Now, I should also point out here that oxygen is required for these processes on these different complexes, and this results in the production of a free radical called superoxide radical. So it's a free radical potentially damaging reactive oxygen species. And cells have ways of detoxifying superoxide and other free radicals. But it turns out, interestingly, some of these free radicals, oxygen free radicals, are actually signaling molecules. I mentioned nitric oxide and relaxation of the blood vessels. Well, nitric oxide is a free radical. It has an unpaired electron, and it can be damaging. It can interact with superoxide to generate something called peroxynitrite, which can cause DNA damage and even cell death if it gets too high levels. So there's enzymes that rapidly remove superoxide, the so-called manganese SOD, superoxide dismutase, also called SOD2. Okay, just quickly, so mitochondria have anywhere from 1,000 to a couple thousand proteins. Only 13 of those proteins in the mitochondria come from DNA in the mitochondria. There is circular DNA in the mitochondria, and it encodes some of the protein subunits of the electron transport chain, but most of the proteins in the mitochondria come from genes in the nucleus that are transcribed there and then translated into the proteins. And those proteins, there's mechanisms for getting them into the mitochondria. Remember, there's two membranes in the mitochondria, an outer membrane, inner membrane. So for example, if you have proteins in the electron transport chain, they have to get across the outer membrane and then become incorporated in the inner membrane. If there's proteins in the mitochondrial matrix inside, they have to be moved all the way across. And there are proteins that have evolved to do this. One thing I wanted to point out is that there are rare genetic disorders that involve mutations in genes that encode mitochondrial proteins. And invariably, the phenotype, that is what's wrong with the individual with these mutations, is manifest in problems in the nervous, muscular system, so nervous system, skeletal muscle system, cardiac cells. These are all excitable cells. And that kind of makes sense because if there's a problem in the mitochondria related to energy production, the cells that are going to be most vulnerable are those that have the highest energy demand and require the efficient generation of ATP through oxidative phosphorylation. So this is just a list of some diseases caused by mutations in genes encoding mitochondrial proteins. A lot of really, and most of these, the problems are evident early in life, during the first few years of life. Okay, so I talked about glucose metabolism. What about fatty acids and ketones? Fatty acids are actively moved into cells, as I mentioned, and they go through a process called beta oxidation to generate acetyl coenzyme A, which enters the TCA cycle, and that can be used in the electron transport chain. I also wanted to point out that neurons can get energy from lactic acid released by astrocytes. So this is pretty interesting. There's this mechanism whereby, if a neuron's very active, if the neurons are very active and lactate levels increase, the astrocytes can provide it to the neurons, and it's actively moved in, and it can be used to generate pyruvate, and that can then go into the TCA cycle and oxidative phosphorylation. Now, there are ways that we can image glucose utilization in brain cells in living people or animals. And the main way that's used is to take radio-labeled glucose, or a form of glucose called 2-deoxyglucose, and you inject it in the intravenously in the individual, and it's low levels of radioactivity, and it's relatively harmless kind of radioactivity, if you will. So it gets into the brain, and this radio-labeled 2-deoxyglucose is transported into cells in the brain. And this 2-deoxyglucose is interesting because it accumulates in the cells because it's not acted upon by this first enzyme in the glycolysis pathway. So it tends to accumulate in cells. And so these are images on the right here showing relative levels of glucose utilization in different brain regions of a normal human being, age-matched person with mild cognitive impairment, and a patient that had been diagnosed with Alzheimer's disease. And what you see is, so red is high levels of glucose utilization. There's less red in the person with mild cognitive impairment and Alzheimer's disease, particularly in the temporal lobe and frontal lobes compared to the age-matched control subject. Right, and so I'll finish up today with this presentation and talk a little bit about ketones, where they come from and what they do. So during fasting or extended exercise, so if you go out and run for an hour, glycogen stores in the liver will be depleted. So this is a hepatocyte, a liver cell in green here. And then what happens is these fats, triglycerides, diglycerides in the adipocytes, the fat cells, are broken down to free fatty acids. They're released into the blood, and they're actively transported into cells, and they undergo beta oxidation to generate acetyl-CoA. And in these liver cells, they have enzymes that then convert this to acetoacetate and beta, or incorporate it into acetoacetate and beta hydroxybutyrate. These are the two ketone bodies that when you hear about whatever, ketogenic diets, fasting, these are the molecules that provide energy to cells when liver glycogen stores are depleted. And so our cells run mainly on these ketones when we're in a fasted state or with extended exercise. So BHB and AA, these ketones, are transported into the blood, out of the blood, into cells. And here I'm showing a neuron in the brain. This is from a review article we wrote a few years ago. So these can be used, there's broken down again to acetyl-CoA and then used to generate ATP. However, these ketones, and I'll talk about this quite a bit later, particularly BHB, is a signaling molecule. It can turn off or possibly, it can turn on or possibly off certain genes. So it can increase or decrease levels of specific proteins in cells through signaling pathways. It can also apparently modify that function of cells, the functionality of cells, by affecting protein function. We found, Christina Morosi in the lab, she found that this ketone BHB in the brain and neurons can stimulate the production of brain-derived neurotrophic factor, which is a very important growth factor that promotes the formation of new synapses, the maintenance of existing synapses. It mediates beneficial effects of exercise, and we think intermittent fasting on learning and memory. It's also important for a process called neurogenesis, which is the production of new neurons from stem cells, which occurs in hippocampus, for example. And then we had evidence that BDNF stimulates mitochondrial biogenesis. So it will cause the growth and division of mitochondria. So when BDNF levels go up, for example, in response to exercise or fasting, over time, number of, actually it's pretty interesting, repeated cycles of exercise and fasting and recovery. So fasting, eating, exercise, resting, sleeping, those cycles over time, say you do that daily or several times a week, over time will increase the number of mitochondria in neurons, increase the number of synapses between neurons, and do other things that seem to be important and beneficial effects of exercise and intermittent fasting on the brain. But I'll talk about that in a later talk. Now, utilization, I mentioned utilization of glucose can be imaged in the brain using MRI, or sorry, using PET imaging, positron emission tomography. The same is true for ketones. And this is an example where Steve Cunan up in Montreal used radiolabeled acetoacetate, the ketone, and he showed when he had the individuals go on a ketogenic diet for several weeks, and he found when they did that, the brain cells switched from using glucose as an energy source to ketones. And I would expect that if we were able to measure BDNF levels in the brains of these individuals, BDNF levels might be up. Another thing he showed is by measuring levels of the ketones in the blood and their utilization in the brain, there's a direct relationship between concentration of ketones in the blood and whether or not, or by how much, the nerve cells and maybe other cells in the brain are using the ketones. Yeah, and then finally, this is my last thing I want to mention today. In this slide, so I mentioned that glutamatergic and GABAergic neurons are the most abundant. Glutamatergic neurons, inhibitory GABAergic neurons. The GABAergic neurons, interestingly, they're more active, at least under normal conditions, than are the glutamatergic neurons. That is, they fire action potentials more rapidly. And that's shown here where recordings from a fast spiking GABAergic interneuron, it's a green cell here, and then a more slowly spiking or firing glutamatergic neuron. And, you know, we and others have done some work showing that there are certain proteins in these very active GABAergic neurons that protect them from any damage caused by the rapid movement of ions in and out, calcium, for example, influx, and the free radicals produced in the mitochondria. So as one example, these GABAergic interneurons have high levels of calcium binding proteins, parvalbumin, calreticulin. This is from a study that Wu Cheng and my lab did, where we had some evidence that these GABAergic neurons degenerate early in Alzheimer's disease, resulting in abnormal hyperexcitability of neural networks and the process we call excitotoxicity, where there's damage to the cells. Okay. Yeah, and so in the next talk, I'll talk about mitochondrial dynamics, how they're moved within cells, how they divide, how they're eliminated from cells, and also talk a little bit about the relationships between activity and mitochondrial activity of neurons, electrochemical activity, and mitochondrial function. But kind of the take-home message there is that when the membrane depolarizes and the neuron fires, calcium moves in to the cell through calcium channels or glutamate receptor channels. So calcium's normally much higher outside the cell than inside the cell. Then that calcium influx, the calcium is taken up by the mitochondria, and that stimulates oxidative phosphorylation. So When neural activity increases, the amount of calcium in the cell increases, the amount of calcium in the mitochondria increases, and that stimulates the electron transport chain. All right, so that's an overview, evolutionary perspective, and kind of basic brain bioenergetics, how the nutrients are used to generate ATP in cells, and how we can get indirect measures of energy metabolism in the brain in living people. So next time, as I mentioned, I'll talk about the mitochondrial dynamics and so on, and then I'll spend a lot of time on effects of intermittent fasting and exercise on the brain. I'll talk about exerkines, factors released into the blood from cells in response to exercise, or fastokines released in the response to fasting. Some really interesting work there showing how our brain is communicated with by our different organs, and how these signals sent from the peripheral organs can affect brain function and resilience. Right, and then the last one I'll talk about aging and neurodegenerative disorders. So I hope you enjoyed this. If you did, or if you have any suggestions, go ahead and put them you can on YouTube type them in to the comments section, and we'll see you next time. Bye.